Good morning, everyone. I'm Dr. Pradeep Kumar Singh, Assistant Professor, Rai School of Sciences, Rai University, Ahmedabad. I'm going to explain about the general structure, properties, and classification of virus. Before going to explain about the virus properties and classification, we will focus on the concept of virus, where the viruses come from and what does it mean. There was the scientist, and that scientist name was the Edward Jenner. Edward Jenner introduced the term virus in 1798 in the microbiology. In Greek word, virus means poisons. Edward Jenner noticed that milkmaids who infected with cowpox developed immunity against smallpox. But how? Then he performed an experiment. He inoculate the vesical fluid which were taken from the hand of the infected maid and inoculated in a boy. Then he noticed after some times the boy developed sustained immunity against the smallpox. Edward Jenner assumed that the vesical fluid that has been taken from the hand of the milkmaid contained a poison, and that poison was responsible for the immunity. Means we can see or we can say a vaccine can be developed from a virus which can be used against another virus. What is the general characteristics of virus? Virus means they are the microscopic microorganisms, means it cannot be seen by our naked eye. What does it mean? Not by naked eye? Means we cannot see by our eyes. Then how can we see the virus? Because viruses are microorganisms, and we know that microorganisms are not seen by our eyes. It can see by the unaided eyes, and that unaided eye is known as microscope. Here is the one question: who is father of microscopes? We can say yes, Antony von Leeuwenhoek was the father of the microscope. He discovered the simple microscopes. He used that microscope for viewing the microorganisms like bacteria. There are the different types of microscopes will be developed time to time. Can virus seen a light microscope? No, viruses can be seen only through the electron microscope because the size of the viruses is very less as compared to the other microorganisms. Means we can see here, we can say viruses are smaller than bacteria. Their range in size between 20 to 400 nanometer, or we can say 20 to 300 nanometer. In some textbooks, the where 20 to 300 nanometer was expressed, and in some other research articles there are the 20 to 400 nanometer, but the range is 20 to 400 nanometer in size. Here is one question from my side: why the viruses cannot keep in any kingdom? What was the basic reason behind this? Rather than all types of microorganisms, all types of cells are kept in a specific kingdom. If you are talking about the kingdom, first of all, there are the two types. There are the two kingdoms. Then after the three kingdom. Then after that four kingdom, and then last one, the scientist R.S. Whitaker described five kingdoms. What are the five kingdoms? First one is the Plantae, second one is the Animalia, third one is the Protista, fourth one is the Fungi, and fifth one is the Monera. These are the five kingdoms. In these five kingdoms, there are the different types of organisms are kept. If you're talking about the animalia, there are the different types of animals, from animals to human beings, are kept in this animalia group, animalia kingdom. Plantae, if you're talking about the plants, plants are the various types like shrubs, creeper, climber, trees. These are the types of plants. They are kept in one kingdom, that is known as Plantae. Third one is the fungi, means all unicellular or heterocellular type of structure of fungus which are saprophytic in nature are kept in fungi. Fungi mainly they are the heterocellular in structure, but some of fungi are unicellular also. Bacteria kept in under Monera kingdoms. There are the two types of bacteria. First one is the true bacteria, that is known as eubacteria. Second one is the extremophilic bacteria, those who can grow in extreme conditions. What do you mean by extreme conditions? Extreme conditions means like extreme temperature, means very high temperature, like 100 degrees centigrade, above 100 degrees centigrade, below minus 10 degrees centigrade. If you're talking about the pH can be 1, pH can be 14, pH can be 7. It can in extreme conditions. Salt concentration, 10% salt concentration. These are the extreme conditions for the growth of the bacteria, that is known as extremophilic bacteria. Then where the viruses are kept in, there is no kingdom. There is no kingdom because viruses are acellular in structures. It is the general properties of virus. Viruses are acellular in structures. Means viruses are non-living cells if it is present outside of the cell. Means what does it means outside of the cells? Means viruses require host cells. Without the host cell, viruses cannot replicate. Viruses cannot be grow. Why viruses cannot be grow outside of the host cell? Because it doesn't have machinery cell. What do you mean by machinery cells? Just I'm taking one example, like me, like you. From the cells makes tissue makes organs makes organ system makes whole body. But for this process, how the functional work will be continue? There are the various systems like digestive system, like respiratory systems. There are the various types of organs are functional. Without this type of organs, we cannot survive. That is known as machinery cells. That's why viruses are non-living and viruses are living when it is present in the host cells. Viruses contain, there is the only one type of genome. We can say genome. That is the one type of nucleic acid. Either it can be DNA or either it can be RNA. But never both. What do you mean by DNA? DNA means deoxyribonucleic acid and RNA means ribonucleic acid. These are the genetic material of the viruses. Viruses consist of nucleic acid, DNA and RNA. It is surrounded by a protein which is known as protein coat, and also it is known as capsid. Some viruses have additional lipoprotein envelope-like structures. Why? Just I told you, it doesn't have machinist cells. Means viruses lack cellular organelles like such as mitochondria and ribosomes. Viruses are obligate cellular parasites. Why am I saying obligate cellular parasites? Why I am not saying facultative? Obligate means it must require host cell. Without the host cells, these viruses cannot be survive because viruses replicate only inside the living cells. Either it can be plant cell, either it can be animal cells, any types, either it can be micro or bacteria, either we can say any type of cells. Viruses replicate means they can make number of copy, they can grow. Viruses replicate through replication of their nucleic acid and synthesis of the viral protein. Viruses do not multiply in synthetic defined media or chemically defined media because chemically defined media is an artificial media, and in that artificial media viruses cannot be grow. Like bacteria, viruses do not undergo for the binary fission. If you're talking about the size of the viruses may be vary, their size may be vary from one virus to another viruses. There are the few terminologies are used in the viruses. First one is the virion. What is virion? Virion is the infected particles. Means it is the complete virus particles. Capsid. Capsid. The nucleic acid is surrounded by a coat that is known as protein coat, and it is known as capsid. What is the nucleocapsid? Nucleocapsid means nucleic acid plus capsid. What is the capsomer? Capsomer is the structure protein units that made up of the capsid. Means capsids are made up from the capsomeres. Not one or two capsomeres. There are the 100, 200 capsomeres are comes together and form a capsid protein. There is the one question will be arise in our mind. What do you understand by defective virus? Defective virus means that virus cannot replicate by its own. It requires helper virus. And it is the unit of nanometer. It is the unit of virus. Means the size of virus will be measured in nanometer. That is 1 nanometer is equal to 10 power minus 9 meter. Either we can say millimicron. Milli means 10 power minus 3 meter. Micron means 10 power minus 6 meter. As I told in the previous slide, virus is composed of nucleic acid. Either it can be DNA or RNA surrounded by protein coat that is known as capsid. And capsid is made up of the small structural units that is known as capsomeres. This capsid proteins protects nucleic acid from inactivation by the outer physical conditions. Like if you are comparing with bacterial cells or any other cells, there are the one structure present outside of the bacteria. That structure is known as cell wall. Like that, like that in virus, there are the protein coat was present outside of the virus. Means the genetic material, the genomic structures was protected, will be protected from the capsid, from the other physical conditions. Few viruses have additional lipoprotein envelope, means we can say viruses are two types. First one is the naked virus or unenveloped virus, and second one is the enveloped virus. If it is enveloped, and that enveloped virus is made up of the lipoprotein structures. Some viruses have enzyme inside the virion. We will discuss about the enzymes in later part of the slides, and that type of enzyme is known as reverse transcriptase enzymes. We'll discuss, just I'm going to explain about the symmetry of viruses. Viruses are divided into three groups based on the morphology of the nucleocapsid and arrangement of capsids. First one is the cubic symmetry. Cubic symmetry means their shape can be spherical in shape, almost spherical like structures, and nucleic acids contained inside the capsid. The icosahedron particle is composed of 20 equilateral triangles. Equilateral means all the angles in the triangles are same side. 12 vertices and 2, 3, 5 rotational symmetry. 2-fold, 3-fold, or 5 in the next slides. That is the 2-fold, 3-fold, and 5-fold. These are the 1-fold, 2-fold. This is known as 3-fold, and that is known as 5-fold cubic symmetry in nature. Second one is the helical symmetry. Helical symmetry means it's a helical-like structure. The virus particle is elongated or pleomorphic in nature, not in spherical way. The nucleic acid is spiral. Capsomeres are arranged around the nucleic acids. That is known as three-complex symmetry. This is the structure of the helical symmetry virus. We can see here. This is the helical-like structure of the virus. If we can see the virus in the electron microscopes, it looks like that. This is the cubic symmetry. The example is the adeno and herpes virus. Helical symmetry like structures, influenza and rabies virus, the example of helical symmetry. Complex symmetry means neither it can be cubic in nature or spherical in nature, neither it can be helical in structures. Their structure is not defined. That is known as pox virus. Complex symmetry. We can classify the viruses into two classes on the basis of their genetic material, on the basis of their genome structures. Those viruses which have DNA, and those viruses which have RNA molecules. That is known as RNA containing virus, and that is known as DNA containing virus. The classification of viruses were described by the one scientist name is known as Baltimore. Baltimore classified the viruses into 6 groups based on their nucleic acid and mRNA productions. These are the 6 groups: double stranded DNA viruses, single stranded DNA viruses, double stranded RNA viruses, single stranded RNA viruses with positive strand, that is known as positive polarity, single stranded RNA virus with negative strand, that is known as negative polarity, Single-stranded RNA viruses associated with the enzyme that is known as reverse transcriptase enzymes. These are the six groups of the classes which are classified by the one scientist. That scientist name is Baltimore and it is known as Baltimore classification. They are the some examples of the double-stranded DNA families of viruses like pox viridase, herpes viridase, hepatodinaviridae, adenoviridae, and papoviridae. Single-stranded DNA families and double-stranded RNA families. Single-stranded DNA family means parvoviridae. Double-stranded RNA family means reoviridae. Single-stranded RNA family with positive strands like picornoviridae, caliciviridae, astroviridae, coronaviridae, flaviviridae, togoviridae. Why the name is ending with viridae? The viral genome acts directly as mRNA. Fifth, single-stranded RNA families with negative strands: Orthomyxoviridae, Paramyxoviridae, Rhabdoviridae, Filoviridae. The viral genome does not act as mRNA. It must be transcribed by the viral enzymes transcriptase into mRNA. Virions contains the enzyme transcriptase. Single-stranded RNA viruses associated with the enzymes reverse transcriptase. Retroviruses, the viral genome is reverse transcribed into a complementary DNA strand using the enzyme reverse transcriptase. Tomorrow we will discuss about the virus replications. How many steps are involved in the virus replications? Thank you everyone. Thank you everyone for listening. If you have any question, then write a mail on my mail ID Pradeep Pradeep dot Singh at raiuniversity.edu. I will try to give answer of all your questions. Have a nice day. Thank you very much.